lots going on. My name is Jobran Chebib. I'm not Chris Marriott, but I'm going to be talking about finding a mate with no social skills. But before I get on with the actual presentation today, I want to talk a little bit about the overarching research project that I've been working on with my collaborator, Chris Marriott. And so I'm at the University of Zurich. I do evolutionary theory and population genetics is my background. And Chris is an artificial life philosopher. That's the kind of work that he does. And we were interested in getting together and trying to simulate emergence in biological systems, especially using agent-based models, to try to get an idea of what is it about the emergence itself, what are the properties that are leading to the emergent systems in biology. So when I talk about the kind of group level behavior that you're going to be seeing in the following slides, I want you to just keep in mind that I am still talking about emergence. So, finding a mate with no social skills. I will start by apologizing to anybody who came to the talk thinking that this is going to help you get a date this evening. Not that it couldn't. It's possible that it could, but our intention was not that. So, the problem that we're going to be working on has to do with sexual reproduction. Sexual reproduction is something that occurs across many types of diverse taxa, which include insects, mammals, and fungi in this particular slide. But as you probably know, we find it across almost the entire tree of life. And one of the problems or the issues that come up when we talk about sexual reproduction is that it requires the co-location in time and space of two individuals in order for that to occur. John Maynard Smith called that one of the costs of sex, and Gimmelfarb called that the social encounter problem, and that's the terminology that I'm going to be using for the rest of this talk. So many people have come up with ideas of how this social encounter problem gets overcome. All of these are social behaviors that overcome this problem. Here are three of them that are commonly cited. One is herding, so you can see here on the left there's a wildebeest that move together on the herds in the plains of Tanzania in this picture. And the basic idea there is that if you spend time with other individuals, the likelihood that you're going to encounter individuals in order to have a sexual reproduction encounter is much higher. The same is true in phylopatry. On the right-hand side, you can see, if you can see it, the sockeye salmon, and they're swimming upstream in the Fraser River there. And again, with phylopatry, you're going back to the same place to give birth that you came from. So in that case, the chances that you meet some other people who are doing the same thing is much higher if more than one person is doing that. So that's another way to overcome this social encounter problem. And the last one on this slide is assortative mating, where you basically look around or smell around or sense around somehow and try to find things that smell, look, sound like you, and you try to mate with them. And that is another social strategy that commonly overcomes this problem. The issue that we get into, especially with people trying to do this in computer simulation, is that this is not as easy as it sounds. I mean, we could encode these kind of things, but it might not be necessary to do so. And the idea in biology that the social behaviors that overcame this problem may have been predated by some other solution to it is that deep in our history, because we've seen this across so many taxa, deep in our history, in our evolutionary history, somewhere our predecessors, long before we had the capability to have these social behaviors, had to overcome this kind of social encounter problem in order to have sexual reproduction. So how did this occur in the first place? And So we wanted to look at is it possible for there to be some kind of non-social behaviors that lead to these same social encounters to overcome this problem. And the way that in order to do that, is we used agent-based models. I'm a biologist, so this slide might seem a little redundant to you, kind of preaching to the choir, but agent-based models are usually good for the type of complex systems like emergence of non-social behavior traits to use in order to simulate them. So let me talk a little bit about our model. The environment itself is a network structure. There's a random connection of nodes, and there is some heterogeneity in the distribution of resources at a particular node. So there are resources at each node, but how many resources you get at each node is different, and the ability to gather resources from each node is different depending on your genes. And then there is also selection for gathering resources, because resources provides you energy in order to survive. So that's the environment. The agents have genes, and each particular gene encodes for a site or a node in the environment, and it tells the agent how to go about gathering resources at that particular site. And there is heterogeneity in how good alleles are for gathering resources. Some take less time and some take more time. And so there will be selection to have certain alleles for a particular site. We have, of course, some kind of variation that occurs. So there's mutation. There's also other types which include crossover or recombination. But the one thing that I want to really impress upon you is that we did not encode social interaction at all in these agents. The agents have no ability to directly detect each other in the environment. So now I talked about the environment, I talked about the genes themselves. Now I want to try to make the connection between the two. I kind of alluded to that a little bit, but the genes encode for gathering resources from a particular environment. So what happens on a daily basis is that an agent wakes up at one of the sites, and he or she looks into their genome and says, where in my genome is there genes for that particular site, so that I can try to gather resources. After it does that, what it does is looks for all current locations, or after it looks for the current locations and finds them, it selects a sequence of sites that are near that site, so that it can move through the network. So it has to be able to have a gene for a particular site, and then it has to look for other genes that are at sites that are connected in the environment, in order for it to be able to move throughout the environment and gather resources. And it tries to do this in some kind of optimal way, where it picks an optimal path that doesn't use up all of the energy that the agent has. So that's the genotype to phenotype map, and it's pretty direct of a connection. Here's some of our simulation parameters. So this was written in JavaScript, and it can run in your browser. And of course, we want to encourage anybody to go and try it out and try the different parameters and see what kind of interesting results you can get out of it. I also wrote it over there. I wrote it twice because I messed it up at the top, but you can see there in case you want to take a look at it later. And this is a little bit about the parameter space, but we basically did 100 replicates of 1,000 days. And what I'd like to actually do is show you what that looks like. So this is actually real time. I'm running it right now. This is a simulation that has So you can determine what the environment parameters is on the left-hand side, and then the population parameters on the right-hand side. And so I just picked some settings just so I could show you what it looks like. And here is a visualization. And what I want you to see basically is for this talk, what's important is that we can kind of in real time we can see the population and how it moves through the network. Where the size of the circle in the network on the very left-hand side determines how many agents are at a particular node. And on the right-hand side, we can see the feeding of the individuals as they feed, and as the circles get darker, the resources at a particular site are being are diminishing, and then once they're black, that means there's no more resources left at that site. And I have a video so that we can get an idea of what it looks like. So you can get an idea of what it looks like when a group is kind of moving in a synchronous pattern together through part of an environment. And that's just a little bit of foreshadowing. So let's talk about the results now. So we have the assortative mating, and what I want to show you here is some graphs where we did a pairwise comparison of individuals. So we took two individuals and we compared their genomes, or we compared their phenomes, and this was basically done by Levenshtein edit distance, so the minimum amount of changes you have to do to get from one to the other. And what I want you to see is at the very bottom here, in these kind of blue ones, we have the dissimilarity metric. So the less similar they are, the two individuals are, the more you'll be on this kind of x-axis, and then the y-axis are all just frequencies. And at the bottom, what we did is we just compared two individuals randomly from the initial population, and we said, how different are they? And we got this kind of, I don't know, Gaussian or univariate normal distribution, however you want to put it, which is what we would expect in a population if they were just randomly. Then what we did is we took the random initial population and we compared them to one of their offspring. And what we see is we can still almost see this kind of normal distribution, but it's kind of melting away as we see that there, of course, as we expect, the parents are more similar to their parents, so there's more individuals that are similar to their parents than are not. So that is something, that's an expected result. And then lastly, what we did is we looked at the final population where we took two random individuals and we compared them, and we saw that overall, both genetically and phenotypically, they tend to be more, the two random individuals tend to be more similar. And this leads back to kind of the sort of mating that I talked about, and this overcomes the social encounter problem that I was talking about at the beginning, because you're basically more likely, if you're just looking around, if you're just trying to mate, the likelihood that you'll find somebody who's more like you, more similar than not, is higher. Now I want to look at phylopatry. So this is the proportion of offspring that are come from phylopatry, or that are phylopathic offspring. So again, we just have proportion and number offspring. And if you look at this final column here on the right, which is the total column, you can see the bottom part is the offspring that come from that are phylopathic offspring, which means that they were born in the same place that their parents were born at. It's greater than 0.6, so 60%. And so that means they're always going. They tend to go back to the same sites that their parents go to, which is what I'm not sure that we'd expect that, because they go to many different nodes in the network. Having the genes that tell them to go to the same site to have kids has led to this kind of phylopatry. And again, it overcomes the social encounter problem, because if you're always going back to the same site that you were birthed at, and other people are doing that same thing, you can find a mate to have sexual reproduction. And lastly, we looked at herding. So what we're looking at here is a heat map where we put 390 agents on the y-axis and the same 390 agents on the x-axis, and we did a pairwise comparison of their phenotypes to see how similar they were after 10,000 days. And if they're very similar, the greatest similarity is really dark, and if they're not very similar at all, then it's white. And then we did a hierarchical clustering analysis on that to see if they clustered into groups. And of course, what we see is these kind of big blocks, big dark squares there, where we see that they're moving together. So again, what we're saying, what we're seeing is we're seeing this kind of herding. It's emerging out of these populations, even though the populations have no way to detect other individuals. Just through having the genetic contingency that leads to other individuals being born and having the connection between the genes and the environment has led to the emergent herding. And again, it overcomes this social encounter problem, because if you're moving in the same spaces, in the same space and time as other individuals, you will be able to find people to mate with. So, kind of the summary is it required no social interaction. There was no direct way for the individuals for the agents to really detect each other, and the genetic contingency was really enough in order to overcome that problem. So, if the genes really encode for something that has to do with the spatial heterogeneity. So the insights that we get from this is that if the social organization like herding, like this emergent behavior that we see, if it can emerge from non-social mechanisms that of just this genetic contingency and the connection between the genes and the environment, well then perhaps social mechanisms themselves, the kind that we see in higher social organisms, where we can actually communicate and interact with each other, might actually evolve from the social organization like herding and sort of mating themselves. And of course, we presuppose that there's probably some kind of coevolution that occurs between these two things. So the next thing we're interested in doing after this is looking for emergence of social mechanisms, so coordinated behavior among agents. So we're going to allow our agents in this kind of simulation to have interaction, to be able to detect each other and see if that leads to the same kind of emergent patterns that we're seeing before. And also, kind of spoiler is we would also like to see emergence occur between the genotype and the phenotype. Currently, we have a very direct connection between our genotype and our phenotype. So our genotype-phenotype map is very simple, but we'd like to develop a genetic architecture that allows for our genes, for our phenotype to really emerge and develop out of our genotype. With that, I would like to thank my collaborator, Christopher Marriott, my supervisor at the University of Zurich, Frederick Bion, and all of our tons of agents who lived and died for this experiment. You can go and look this up. You can find us on ResearchGate or LinkedIn. And if you see us around the conference in the next couple of days, please feel free to come up and talk to us. We're really interested in talking to anybody about emergence, and we're really interested to talk to anybody about any kind of collaborations, because we really think that getting interdisciplinary specialists to help us build these kind of things is important. Thank you.